So to start the discussion, we have a team of experts from the International Rice Research Institute, or IRRI, who will talk about the importance of the gene bank and will give us a virtual tour. And I will be giving you the IRRI team of experts, the name of the IRRI team of experts who will be joining us today. Ma'am Eileen Garcia, the manager or the project coordination and stakeholder advocacy, Healthier Rice Program. Selfa Marie Bombais, Marionette Alana, Norman Oliva, Ana Maria Reynoso, Amiri Amparado, and Dr. Benu Prasad Ramaya, the research unit leader of Future Genetic Resources at IRRI. So, actually, the tour is one of the activities in Farmers Exchange Program that we conduct every year to help our participants become familiar with the importance of gene bank in crop improvement and importance of seed quality. So IRRI team, the floor is yours.
Good day, everyone. Can you hear me well?
Yes, ma'am. Yes.
Yes. Okay. Good day, everyone. On behalf of the Healthier Rice Program of the International Rice Research Institute, it is my pleasure to welcome you all to this afternoon's activity of the Pan-Asia Farmers Exchange Program. My name is Anna. I will be your host and moderator, and I will be supported by my IRRI colleagues as our virtual tour progresses. As you can see on your screens, we have an aerial view of the International Rice Research Institute campus, and it was founded in 1960 by two American foundations. We have the Ford Foundation and the Rockefeller Foundation in partnership with the Philippine government. So Los Baños, Laguna, Philippines was the chosen site among other rice-producing and rice-consuming regions in Asia. Well, due to its proximity to the University of the Philippines, Los Baños, or UPLB, which back then was the premier agricultural university in Asia. As a matter of fact, the IRRI headquarters sits on a 200-hectare land, which includes an upland and lowland experiment stations, which is located next to the university. To date, our workforce is composed of about 1,000 staff from 37 countries. And while our headquarters is based in the Philippines, we do keep our collaborations robust and our international presence strong through our 16 country offices sprawled across Asia and East Africa. However, due to the COVID-19 pandemic, the Institute has been operating at a limited capacity, and because of this and other travel-related restrictions, we are unable to receive visitors at the moment. I am pleased to inform you that our team has put together a very special virtual tour of IRRI to take you through the facilities that have been instrumental in taking us on the road to golden rice. So our tour will focus on three facilities. We have the International Rice Gene Bank, the Genetic Transformation Laboratory, and the Transgenic Screenhouse. So at each station, we will be joined by a resource person. And then towards the end of our tour, we will have a designated time a designated discussion period to answer questions that you may have. So I would just like to remind everyone to please send your questions through the chat box on this meeting. Another reminder, most of the photos and video materials that you're about to see, these were recorded pre-pandemic. So you might notice the absence of mouth covering or physical distancing, and it's because of that. During the previous years that we welcomed the participants of the Farmers Exchange at IRRI, this is how we did the tour. We have our participants in a trailer, and this is driven by a tractor, and then we drive around 300 meters to our laboratory. So we will now proceed to our first site, to the International Rice Gene Bank. Please welcome my colleague, Ms. Monette Alana.
Good afternoon from the International Rice Gene Bank, or the IRRI Gene Bank, located in the Nile Sea Brady Laboratory. I am Winette Alana, Associate Scientist, Gene Bank Seed Management. I will help you navigate this important resource center at IRRI. The IRRI Gene Bank is the world's biggest collection of rice genetic diversity in the world. It holds 132,140 accessions of rice as of September 2021. This includes cultivated species of modern rice, traditional varieties, and wild species. Countries from all over the world send their rice seed samples to IRRI for safekeeping and for sharing for the common public good. It is an essential resource for food security, as the conserved germplasm is used to develop and improve new varieties that are higher yielding or are more resistant to pests, diseases, and stresses of climate change. Established in 1971, the IRRI Gene Bank and associated laboratories, offices, and supporting facilities are over 40 years old, but has been operating at the highest approved international standards. Here are the current figures of some of the contributions to the Gene Bank. An exact copy of IRRI Gene Bank's collection is stored in the Svalbard Global Seed Vault in Norway. The most recent IRRI deposit, the Svalbard Vault, was made in February 2020, just before the pandemic, by Dr. Benu Maya, Research Unit Leader of Feed for Future Genetic Resources at IRRI. If you have questions on this, Dr. Benu is with us later in this session and will be happy to answer questions you may have. Traditional varieties and wild species of rice are being lost through genetic erosion. Farmers adapt new varieties and stop growing varieties that they have used and nurtured for generations, and eventually lose these varieties. Wild species are threatened with extinction as their habitats are destroyed by human disturbance. Future crop improvement needs genetic variation from traditional varieties and related wild species to cope with the many biotic and abiotic stresses that challenge rice production around the world. By collecting and conserving rice germplasm, this is how IRRI Gene Bank safeguards genetic diversity and improves crops. With access to the world's largest collection of rice, we have a unique opportunity to study the diversity of rice. Scientists characterize rice for its traits and genetic makeup to find useful versions of genes and determine the best method of improving the rice variety, whether through conventional breeding or more precise breeding techniques like genetic engineering or gene editing. In the case of golden rice, genetic engineering was used because the screening of rice germplasm showed that beta-carotene is not present in traditional rice varieties. Each type of rice in the IRRI Gene Bank is stored in both the base and active collections. The base collection is used for long-term storage and maintained at -20 degrees Celsius, while the active collection stores seeds available for distribution at 2 to 4 degrees Celsius. At IRRI Gene Bank, rice seeds have the potential to remain viable for many decades. Viability testing of all samples is carried out every 5 years for seeds stored in the active collection, or every 10 years for seeds stored in the base collection. If viability falls below 85%, samples of the remaining seeds are planted to produce fresh seeds for storage. IRRI shares seeds of small quantities free of charge on demand to any individual or organization anywhere in the world for purposes of research, breeding, or training for agriculture. Because of the limitation of seed multiplication during this pandemic period, only limited seeds are available for distribution. As staffing and operations increase at a higher capacity, more seeds will be available for sharing. Over to you, Norman, at the Genetic Transformation Laboratory.
Hi, I'm Norman Oliva, Senior Associate Scientist at the Genetic Transformation Lab, or GTL. GTL is central to our work on genetic engineering. The lab is instrumental in conducting the molecular, characterization, and safety assessment dossier used in the regulatory application for golden rice. It is also leading the work in high iron and zinc rice. I hope you can recall Dr. Russell Ranke's presentation last Monday about golden rice, where he discussed the journey of golden rice from its early stages, its development over the years, and final approval in selected countries. Biofortification is a process of nutritionally improving rice. Golden rice has been biofortified through genetic modification. I shall now show you the toolbox for developing genetically engineered rice for improved nutrition. The first step is gene cloning, in which the gene of interest is replicated in preparation for transfer. DNA or RNA is extracted from the organism, and the target gene is isolated by PCR technique. Next is agrobacterium transformation and the tissue culture process. The gene of interest is combined with the appropriate rice promoter to regulate the gene expression and inserted into the agrobacterium PI plasmid. The expression vector for agrobacterium transformation of rice is analyzed by restriction analysis and sequencing. Immature rice embryos are co-cultivated for 7 days with the agrobacterium containing the gene of interest. Once they have sufficiently germinated, transgenic cell lines are selected and regenerated into plant. This can take up to 70 to 75 days. Transgenic plants develop from separate single cells, or what we call independent events, are grown hydroponically for 2 weeks and subjected to PCR molecular analysis for the presence of the gene. Plants are transplanted to grow into maturity. Normal and fertile plants are selected for further molecular analysis of single copy gene and gene expression. The off types, or the ones that do not grow to maturity, are discarded following strict biosafety protocols. Once the plants are mature enough, they are transplanted in our transgenic Screenhouse facility, which you will see shortly. IRRI is accredited by the Excellence in Stewardship Program, a global organization that promotes adoption of product stewardship programs and quality management. The Genetic Transformation Lab was responsible for conducting the molecular characterization and safety assessment tests for the regulatory applications of golden rice. These results were published in Nature Scientific Reports in 2020. Internationally accepted standards for the safety assessment of genetically engineered food crops involve the molecular characterization of the inserted DNA and an evaluation on the allergenicity and toxicity of the newly expressed proteins from the genetic transformation. A complete analysis indicates that the inserted DNA was introduced without modification and the trait was stably inherited across multiple generations. Comparative analysis and laboratory tests demonstrate that the new proteins in the GR2E golden rice grains are as safe as any existing proteins in ordinary rice. This study supports the conclusion made by regulators in New Zealand, Australia, Canada, the United States of America, and the Philippines that food derived from golden rice is as safe as food derived from conventional rice. Over to you, Amory. Let me turn you over to Amory, our Screenhouse facility resource person.
Good afternoon, everyone. The last facility we are visiting in the is the IRRI Transgenic Screenhouse, which is used for confined testing activities and for integration of golden rice into popular local rice varieties. Good afternoon, I am Amory Amparado, your assistant scientist in the Healthier Rice program at IRRI and your Transgenic Screenhouse resource person. The Transgenic Screenhouse is where we grow genetically engineered rice varieties like golden rice. In previous years, we've conducted confined tests as part of the biosafety regulatory process. This was done to demonstrate that golden rice is as safe as ordinary rice, with the added benefit of beta-carotene in its grain. Our studies show that agronomic, yield, and grain quality traits of golden rice are the same as its ordinary rice counterparts. To bring golden rice from proof to concept to a market variety, scientists and breeders at IRRI, Philippine Department of Agriculture, PhilRice, and the Bangladesh Rice Research Institute introduced the beta-carotene producing GR2E golden rice trait to popular inbred rice varieties through marker-assisted backcross breeding. These days, we are growing golden rice to produce the seeds that will be used in the pilot scale deployment. And like you, we are very much excited to see golden rice move closer and closer to the people who need it to the most. Back to you, Anna.
Thank you, Anne-Marie, Norman, and Monette. We hope that this virtual tour has added to your knowledge of golden rice and the process of biofortification. If there are some points that need clarification, we have IRRI scientists in this session who will be happy to take your questions. So another reminder, if you have comments or questions, please send them through the chat box in this meeting. And apart from our three resource persons, we also have in attendance Dr. Venu Prasad Ramaya, Research Unit Leader of Future Genetic Resources at IRRI. Thank you for joining us, Dr. Venu.